Hi, my name is Lasse, and I'm a PhD student at the Center for Evolutionary Hologenomics at Globe Institute. Hologenomics to me is like putting on a pair of glasses that allows you to see the complete genetic machinery inside a host organism, looking both at the genome of the host as well as the genomes of all the microorganisms living inside the host. And without these glasses, we're only seeing what's going on the surface. Originally, I wanted to do population genetics on fish, but then I was presented with the possibility to combine this with the study of the genomes of all the microorganisms living inside of fish. And this allowed me to do what I originally wanted to do, but at the same time be a part of the ongoing development of this new and exciting field that is hologenomics. I work within the field of conservation hologenomics. I studied the microbiome of different populations of two highly endangered fish species living in the wetlands of the Mediterranean Sea. I compared the microbiome of these wild fish to individuals of the same species that are bred in captivity. Our hypothesis is that fish bred in captivity will lack certain functions that are normally provided by the microbiota in wild fish, and that this will have consequences when fish bred in captivity are released into the wild. My approach is different from previous work in that I seek to implement the hologenomic approach in an applied conservation setting. And in order to do so, I first need to establish the microbial differences between fish bred in captivity and our wild counterparts. Once we establish these differences, we need to come up with a way of engineering the microbiome of fish bred in captivity in order for it to look more like the microbiota of wild fish. The next step will be to test if this engineering makes a difference when the fish are released into the wild. And the way we will test this is by taking fish bred in captivity with a wild engineered microbiome, release them into the wild, and compare their survival and fitness to fish bred in captivity with a normal captive microbiome. When I started my undergraduate in biology, I had a firm belief that I wanted to be a hardcore field biologist and never work in a lab. But through the years, this has changed completely as I have realized all the possibilities provided by molecular biology. So today I do both field work, lab work, and data analysis, and I feel like I have the perfect balance between the three.